I will discuss the characteristic features of very common fungi belonging to Ascomycetes and Basidiomycetes and others now. In Ascomycetes, the somatic body is composed of highly branched, well-developed, branched septate hyphae. Although unicellular members are also present in this group, for example, Schizosaccharomyces or fission yeast, Saccharomyces cerevisiae, budding yeast like members. Some of the members exhibit dimorphism, as you can see here. In septate version, the septa is always provided with a simple pore, and in dimorphic fungi, you will find unicellular form along with the pseudohyphae produced by their aggregation or rather connection. In septate form, the simple porous septa remain associated with some membrane-bound organelle called oronin body, inside which the crystalline amorphous protein remain present. These oronin bodies may be spherical, hexagonal, rectangular, cylindrical, and so on. Besides oronin bodies, in Ascomycetes, the septal pore remain blocked or plugged with some pulley wheel-like material, usually termed septal pore organelles. In this group, cell wall is mainly composed of chitin and some short fragments of cellulose. However, in unicellular form, the cell wall is composed of mannan and beta-1,3-glucan. Very small amount of chitin is present and it is restricted mainly in the bud scar. During asexual reproduction, mitospores are usually produced, which may be conidia or chlamydospores like structure, but the chief method of asexual reproduction is fission or budding or fragmentation. During sexual reproduction, two nuclei from two different parentage must come within a cellular compartment, and it is achieved with the help of fusion between two similar gametangium in yeast, as you can see. Fusion may be between two dissimilar gametangia, well differentiated one, that is Ascogonium and Antheridium. Spermatization is another means of sexual reproduction where a very small thin-walled male cell come in contact with the receptive hyphae and they fuse with each other to form a dikaryotic structure having a cell with two different nuclei in each compartment. Now, somatogamy is very simple type of reproduction where two normal hyphae fuses with each other and make a hyphae dikaryotic. Whatever may be the procedure by which two different nuclei of two different parentage come in each cellular compartment, this dikaryote phase is very much short-lived and parasitic on haplophase mainly. The spores produced by this group of fungi is called ascospores, which are produced in a sac-like structure called ascus, although globular ascus is also common in this group. Within ascus, usually 8 ascospores remain confined. That means ascospores are endogenous in nature. These ascospores containing ascus are present in a definite layer in some type of fruit body, particularly in apothecium. The layer within which the ascus containing ascospore remain arranged in a palisade-like manner is called hymeneal layer. Within hymeneal layer, the paraphyses as well as ascus containing ascospores are present. The ascocarp are the main macroscopic structure produced by the Ascomycetes fungi may be Cleistothecium. It is a globular structure without any natural opening. Perithecium, it is a flask-like structure with a prominent and dominant ostiole at the apex, and Apothecium with wide open hymeneal layer. Now, within Ascomycota, three different subgroups are recognized: ARCHIASCOMYCETES, HEMIASCOMYCETES, and EUASCOMYCETES. In ARCHIASCOMYCETES, usually members like Saccharomyces pombe, Taphrina deformans, Protomyces like primitive members, and plant pathogens are included. Members of this group of organism are characterized by absence of ascocarp during their life cycle. They produce naked asci from either individual yeast-like cells or by the conversion of their hyphal tip. The chief mode of asexual reproduction is fission or budding. The ascus cavity, say for example in Taphrina deformans, remain in a heterogeneous form where one can find ascospores along with budding ascospores. That means within an ascus of Taphrina deformans, ascospore and a mixture of budding ascospores remain present. Cell wall contains very little amount of chitin, and that only restricted in the bud scar. Now comes to HEMIASCOMYCETES. In this group, the budding yeast, that is Saccharomyces cerevisiae, are included. These are basically unicellular organism, but sometimes they produce pseudomycelia. The main characteristic feature of HEMIASCOMYCETES are presence of their prominent growth form unicellular or yeast state. Cell wall contains very little chitin. One or several pores may be present in their septa, which may be very small or may be plugged. They lack oronin bodies, which are one of the important characteristic features of EUASCOMYCETES. In EUASCOMYCETES, the macroscopic ascocarp producing members are included. You can see here the ascobolus, alluria like members, and this group of organism is characterized by their highly branched, well-developed septate hyphae. Septa is usually provided with one large pore associated with oronin bodies and septal pore organelles. Ascocarp are always produced after their sexual reproduction. Now, from phylogenetic study, it is apparent that the Ascomycetes member and Basidiomycetes member are the sister groups, both of which are derived from Chytridiomycota after loss of their flagella. Within this particular Ascomycetes group, ARCHIASCOMYCETES present the most primitive type of Ascomycetes fungi. That is, they diverged very early during the course of evolution, and after that HEMIASCOMYCETES and EUASCOMYCETES are derived. This ARCHIASCOMYCETES is usually monophyletic, but EU and HEMIASCOMYCETES are polyphyletic in nature. Now comes to Basidiomycota. In this group of organism, the somatic hyphae is well developed, profusely branched, and septate one. The septa are always provided with a typical swollen pore called dolipore, and the pores may be guarded by or remain covered by a cap-like structure called pore cap or dolipore cap or parenthysome. Now, the parenthysome may be perforated or non-perforated or imperforated one. Cell wall in this group composed of mainly chitin and beta-glucan. However, unicellular yeast-like members of Basidiomycota cell wall is composed of chitin and mannan mainly. During their life cycle, members of Basidiomycota produce both monokaryotic and dikaryotic hyphae, and within a particular food body one can find three different types of mycelia: primary, secondary, and tertiary. Primary mycelia are always monokaryotic and homokaryotic and are produced from the germination of basidiospore as such, but dikaryotic hyphae are always provided with a typical clamp connection or bypass, which makes the entire hyphae dikaryotic. Meiosis occur within the basidium, and that's why basidium is considered as the sex organ or sexual apparatus of Basidiomycota. After meiosis, four distinct nuclei are produced inside the basidium, which ultimately enters into the four basidiospores produced on the tip of the basidium. Thus, basidiospore remain attached with a very stalk-like or peg-like structure called sterigmata. Within Basidiomycota, three different subgroups are recognized: Uredintomycetes, Ustilaginomycetes, and finally the Hymenomycetes. In Uredintomycetes, the septa may be provided with uniperforate simple septa, or the septa may be plugged or may remain open, but dolipore septum, the characteristic of Basidiomycetes, is absent completely. Both primary and secondary hyphae grow intercellularly and produce distinctive hostoria, the sucking organ, inside the cell. The members of Uredintomycetes never produce basidiocarp, the macroscopic structure of Basidiomycota. The karyogamy takes place within a specialized pore called teleospore, which is considered as a probasidium. So, inside the teleospore or probasidium, karyogamy takes place, but meiosis occurs inside the promycelium or metabasidium. The metabasidium are always transversely septate and produce four basidiospores laterally. In Uredintomycetes, 5 different stages occur in their life cycle, of which stage 0 represented by the spermatia and spermogonia, both are haploid in nature. Stage 1 is represented by acial cup bearing dikaryotic aciospore. Stage 2 is represented by Uredinia bearing dikaryotic uridospore or uridinospore. Now, stage 3 represent the tilia or tilial cup bearing dikaryotic tidiospore, within which the karyogamy and meiosis takes place. Now, during germination, these teliospores gives rise to a short four-celled promycelium on which the basidiospores are produced laterally. However, not all these 5 stages mentioned now occur in a same species, and accordingly the members of Uredintomycetes are classified into macrocyclic rust, microcyclic rust, or demicyclic rust. Now comes to Ustilaginomycetes. Members of this subgroup are mostly parasitic on higher plant with a free-living saprotrophic capacity. The dikaryotic hyphae are mostly parasitic on higher plant, as you can see in the picture. However, members of Ustilaginomycetes are dimorphic, that means they can exist in 2 particular growth form. One is hyphal, septate hyphal form, and another one is budding yeast-like form. Usually, the basidiospores or the unicellular spores produced by them buds out and constitute the unicellular budding phase or stage. The hyphae of Ustilaginomycetes is usually without any perforation, but their septa may be provided with a very small simple pore. Whenever dolipore is present, it is devoid of any septal pore cap or parenthysome. Members of Ustilaginomycetes usually produce a clump of typical spores called smart spore, chlamydospore, teleutospore, brand spore, etc. These spores are dikaryotic in nature. Within this dikaryotic spore, karyogamy takes place and thus it behaves like a zygote. On germination, these chlamydospores or teleutospores gives rise to numerous basidium instead of usual 4 basidiospores. The third group recognized under Basidiomycotina is Hymenomycetes, which is characterized by the presence of typical hymeneal layer composed of club-shaped basidia bearing basidiospores and the paraphyses. Usually, this hymenium remain exposed or become exposed with the maturity of the basidiospores. Now, if you study the phylogeny of Basidiomycota, you will see that 3 different groups are recognized: Uredintomycetes, Ustilaginomycetes and Hymenomycetes. In Uredintomycetes and Ustilaginomycetes, no basidiocarps are produced as such, but typical basidiocarp is produced by the members of Hymenomycetes. So, these 3 groups are considered as the sister group of Ascomycota, since these are the members of the typical phylum Basidiomycota. The mitosporic fungi. You know that mitosporic fungi reproduce asexually always, that is, sexual reproduction is either absent totally in this group, sex organs are not reported so far in this group of fungi, and they mainly reproduce by conidia like unicellular non-motile spores which are differentiated asexually. As in Penicillium, the conidia are produced exogenously. In Trichoderma viridi, the conidiospores are again differentiated externally. Whenever their sexual reproduction is known, these members are transferred to their respective group depending upon the nature of their sexual spores. For example, members of Penicillium, after discovery of their ascospore, known as Talaromyces, transferred to Ascomycetes. So, in mitosporic fungi, members of Ascomycetes and Basidiomycetes are included, which reproduce absolutely by mitospore, and sexual reproduction is either absent or unknown so far. Now, I will concentrate on the kingdom Straminiphylla. This group of organisms represent a diverse group with heterocont flagella, and it is believed that they are the diverged from the group called heterocont algae. Within the kingdom Straminiphylla, the phylum Umycota is recognized. The plant body of Umycota includes both unicellular, holocarpic or eucarpic filamentous somatic body, but they are always diploid. Like other chytrids, they are either eucarpic or holocarpic in nature, and based on their life cycle similarity and similarity with their somatic structure, the members of Umycota are earlier placed under Chytridiolis order or under Chytridiomycota. They are always diploid, however. The cell wall is composed of cellulose and glucan mainly. Cell wall protein is rich in hydroxyproline, but in true fungi, proline is the dominant protein amino acid. In Umycota, mitochondria is with tubular cristae and Golgi body is with multiple flattened cisternae. In Umycota, a typical organelle or vacuole is present with paracrystalline electron-dense lamellar structure which are very much similar to the fingerprints, and that's why such vacuoles are called electron-dense or dense body vacuole or finger vacuole or fingerprint vacuole by different mycologists. Within this vacuole, paracrystalline lamellar array like structure are present resembling fingerprint. In course of time, this paralamellar structure, paracrystalline structure condense to form a globular or vesicular structure. This is one of the characteristic feature of this group of fungi. This particular vacuole actually contains the mycolaminarin or mycolaminarin phosphate inside. Asexual reproduction takes place by means of flagellated zoospore, and flagella is of two different types. Whipless flagella is very shorter, but tinsel flagella is very longer one. During sexual reproduction, two different types of sex organs, that is oogonia and antheridia, are differentiated, and fertilization tube, you can see, play a role in the process of fertilization. The resultant spore is always oospore, which is diploid in nature, and this diploid spore usually germinates to form diploid sinusitic plant body of Mycota. Now comes to the Hyphochytridiomycota. In this group of organisms like Chytridiomycota, they may be holocarpic endobiotic, they may be eucarpic epibiotic, they may be eucarpic polycentric, etc. In holocarpic form, what happens? After a brief period of sexual reproduction, they produce a resting sporangium or resting spore, within which the spores remain confined as such. In epibiotic form, the small rhizoidal part radiates from a particular point, but in polycentric one, the resting sporangium remain connected with a branched hyphal structure being composed of branched hyphae. This is a photograph of a eucarpic polycentric thallus of Hyphochytridium. Cell wall in this group is composed of chitin and cellulose mainly. Reproduction takes place by means of a uniflagellate zoospore. The zoospore is always anteriorly uniflagellate and hence provided with a tinsel flagellum. After a brief period of swimming in the surrounding water, it loses its flagella, encysted, and the encysted zoospore germinates to form the plant body, as you can see here. The unicellular plant body may be with rhizoid, without rhizoid, may be epibiotic, may be endobiotic, or may be polycentric, as I have said earlier. The next group of organism under Straminiphylla is the Labyrinthulomycota. This group of organism is peculiar in that sense that their somatic body is very much spindle shaped, provided with a typical surface organelle called bothrosome or sagenogene. These bothrosomes are produced de novo during their asexual reproduction or during cell division. This bothrosome, as you can see, that is a small structure which usually remain plugged by a dense cytoplasmic material. This spindle-shaped plant body is aerodynamically so sound that it can freely move inside the ectoplasmic net or slime net produced by the individual cells during their course of growth. These spindle-shaped cells actually exhibit gliding motility inside their net or called slime net. This motility or movement is essential for their cell-to-cell communication, for their adhesion, as well as for nutrition. Now, the ectoplasmic network secreted or synthesized by the individual somatic cell consists of two membranes. One is outer membrane and another one is inner membrane. At bothrosome, This inner membrane is continuous with the plasma membrane. In this picture you can see this is the plasma membrane and this is the inner membrane of the ectoplasmic network. This particular junction remain blocked by a dense cytoplasmic plug in order to prevent movement of cellular organelles into the ectoplasmic network. The typical cells of labyrinthids are composed of Golgi-derived ultra-thin scales. It is a polymer of L-galactose. However, in members like Cleistothecium, the cell wall is composed of Golgi-derived ultra-thin scales, but bothrosome present at the base of the globular body and the junction between rhizoidal structure. Cleistotheciums are usually monocentric epibiotic body. Now, from these diagrams you can see that how colony of the labyrinthids looks like. These are the spindle-shaped somatic body and these net-like structures are produced by the individual somatic cells and are designated as either ectoplasmic net or slime net. Asexual reproduction in this group takes place by simple transverse cleavage and rotation inside the ectoplasmic network. Sometimes they also produce wall-less zoospore which are biflagellated in nature, provided with anterior tinsel flagella which is long one and a posterior short whipless flagella. However, in the members which reproduce sexually are equipped with a typical small eye spot, and this eye spot of labyrinthid zoospore are active phototactically. Placement of Mycota, Hyphochytridiomycota and Labyrinthulomycota is justified under Straminiphylla, and it is believed that these three groups of organisms are more closely related to heterocont algae in comparison with the true fungi discussed so far earlier. Thank you.